welcome everyone. Welcome back to the first session after the summer break of the Neuropsychiatric Working Group. I hope you all had a great summer, you all were safe, and it's great to be back. For housekeeping, we made a few changes to the format. So we decided to meet every 2 months for the working group. And we still have the monthly evening lecture. I hope you stay on. Tonight we'll have Elise Robinson. If you're interested, Jonathan, we can, if you don't have I think you are on the mailing list, but if not, we can send you a link. Elise Robinson is giving a talk after you at 5 o'clock. And I also meant to ask you, because I always forget, will you permit us to record your remarks and may read them, post them? Okay, great, thank you.
Of course, and I'm sorry that I can't attend tonight.
Yeah, well,
I have meetings all the way through.
Okay, I figured, yeah. So just a few updates before we invite Jonathan. So the activities of the working group are ongoing. We have now 3 projects that I hope I can tell you more in the next few months that will go in as grants, coincidentally, to the National Institute of Mental Health. 2 will be on functional genomics. I think when we mentioned them briefly before, in collaboration with Columbia, the Scripps, UCSF, yet all coming out of the working group. And then the other one, of course, is our precision psychiatry cohort, the GEMMA project, you'll know as the GEMMA project, that we have re-christened as COPPA, which is a which we'll hope will serve as a proof of principle for the larger platform, a precision psychiatry based project that will focus on psychotic disorders. But again, all of this, you'll hear much more and in more detail in the next few months, but I just wanted to make sure you are in the loop. Lastly, for introduction, I hope that you will continue to join us. I hope that you will nominate speakers for the evening lecture, that you also nominate yourself to talk to us about your work in the working group sessions. So since we only meet every 2 months, so we have about, 6 before the end of this season, which will be in June of 2022. So with having said this, it is a great pleasure to welcome Jonathan Pevsner as today's speaker for the working group session. Jonathan is the new branch chief of genomics at the National Institute of Mental Health. I've asked him to tell us about his vision of psychiatric genomics, and he has graciously agreed to do so. Many of you will know him from his work in mosaicism and functional biology of neuropsychiatric disorders in general. He came to the National Institute of Mental Health from Johns Hopkins, where he was a full professor. And he has the job now that I had many years ago. So I'm particularly delighted to have Jonathan here, and I want to welcome him. Please take it away and share your wisdom with us.
Thanks so much for inviting me and for that introduction. I'm not sure about the wisdom, but I'll do my best. And so I'm going to start by sharing screen and just check that this is working okay.
It's not on full screen yet. So yes, now we can see the full screen.
Great. And so my disclaimer is that some of this work was conducted while I was employed at Kennedy Krieger and Johns Hopkins School of Medicine. And so I have learned that joining the government, one needs to check on disclaimer slides like this. An outline of what I'd like to present is to briefly talk about the human genome features and sequencing, briefly talk about comparative genomics from really a historical perspective, survey the report of the Council Workgroup on Genomics that Thomas had a leading role in developing. And indeed, much of what I have to say really comes from Thomas's vision and initiative. And so it's just a pleasure to come into NIMH, where his impact has been so enormous within NIMH and across the whole field. And then I'll end with a discussion of rare and common disease with two diagrams. One extends from genotype to phenotype. The other one is a plot of allele frequencies and effect sizes. And so beginning with the human genome, of course, as our DNA is organized into chromosomes, I want to remember the beautiful structure and function of the chromosomes, in this case is shown by Darlington, and think about genomic DNA in terms of chromosomes and in terms of chromosomal abnormalities, such as in this case, a 11q deletion in a patient with intellectual disability and other conditions. And so it's now become routine to sequence genomes. And when we do so, we find different kinds of variation, such as if we sequence one genome, we might find on the order of 4 million single nucleotide variants and 600,000 structural variants. And then of our 20,000 protein coding genes, we might find on the order of 11,000 non-synonymous variants in an individual's genome. Perhaps 100 or 200 are disease associated based on previous annotation of the genome, and several dozen may be knockouts. And so as we look at this, I want to say that I'm very interested in technologies that can be applied to understand the genome. And one of them is really a landmark that was just announced several months ago. The Telomere to Telomere Consortium completed the first whole human genome with assembly, in this case from a homozygous high datidiform mole cell line, which is homozygous, making it easier to sequence, but it's the first complete genome that goes end to end without gaps, with just over 3 billion base pairs. This is a high resolution string graph built from PacBio hi-fi reads. It's gapless and it covers the autosomes and the X chromosome, but no Y yet, because this was a female derived line. And it has the mitochondrial genome, and it adds, I think, about 8% of the known sequence to the genome, with 238 megabases compared to GRCH38, mostly centromeric, some many segmental duplications, resolving ribosomal DNA clusters that have been too challenging to solve up till now. And it adds several thousand paralogs, including 115 that are predicted to be protein coding. My first thought is whether any of those newly discovered paralogs might be implicated in neurodevelopmental and psychiatric disease. And so my last slide on this section is to talk about these sequencing technologies and to say many people do short read whole genome sequencing with 30x average depth of coverage. And that's very good to reveal single nucleotide variants, indels, and structural variation to a certain extent. Long read sequencing does a much better job at detecting the structural variation. And most people are not doing it now because it's so expensive relative to short read sequencing. And yet one wonders whether there are structural variants that are recurrent and even penetrant that are relevant to autism spectrum disorder, schizophrenia, and other conditions. And this remains to be seen as the first cohorts become sequenced. Now, many people who are applying for NIH grants have a real tension or challenge given a particular limited budget. What is the sample size they can achieve with particular sequencing technologies? And so low-pass whole genome sequencing has been emerging, for example, as an option, for example, 4x or even below 1x average depth of coverage, which provides great cost savings, but has less sensitivity to detect heterozygous variation in particular, or rare variation. And so this is a constant debate about what to do. Another alternative is whole exome sequencing. And here, this is extremely popular, and we're probably in the millions of exomes that have been sequenced. And yet, it's a very different kind of look into the genome and what is thought to be the functionally most relevant part. And I'd like to mention there are ongoing efforts to develop collections of high quality, complete reference genomes of diverse ancestry, which will again help in genotyping as ancestral populations are sequenced more and more frequently. And even the Telomere to Telomere recently introduced consortium, Telomere to Telomere Consortium recently introduced reference genome is a better reference to use than GRCH38 when looking at whole exomes. And so this is a very actively developing area. My next topic is comparative genomics and evolution. And I can think of three main reasons why we might want to do comparative genomics. to study genomes for their own sake. Second, to understand what makes our human genome unique. And we compare our genome to those of primates and other organisms across the tree of life. And then a third goal is to understand variation across human genomes, comparing genomes derived from different geographic origins and across diagnostic groups. And so I'd like to show this graphically. This is a figure actually from a textbook on bioinformatics that I wrote from 2015. And this is showing human and chimpanzee next to mouse and rat, dog and chicken, and fish. And at the nodes, we have the last common ancestor, which might have lived 5 to 7 million years ago for chimpanzee, 80 million years ago for the rodents, going on back to 300 million years ago for the birds, and over 400 million years for fish. And so if we compare the human genome to the fish and we find identically conserved sequences, these may have particularly important functional properties. And similarly, we can look at gene conservation across the vertebrates in this case, or further across the tree of life, to understand what might be important. Then within human populations, what's sometimes called population shadowing allows us to compare genomes from different diagnostic groups. And so I'd like to take a moment now to take a deep dive into history. And I'm going to start with 1838, which was the year that Mulder said, he wrote, the egg white-like bodies, albumin, have occupied a considerable part of my life. All kinds of difficulties had to be surmounted, difficulties not met with other bodies. Whatever may have been said about this, one point is certain, that I've been the first who has shown in 1838 that the meat is present in the bread and the cheese in the grass. What he meant by this is that he was one of the people in the early 1800s who contributed to the initial discovery of proteins. And he recognized some of the molecular formulae that underlie proteins. He recognized some of the amino acids that comprise proteins, and seeing proteins in both plants and animals, he was recognizing here the deep connection between life forms through protein. So that's why he wrote this amazing phrase, the meat is present in the bread and the cheese in the grass. That the whole organic kingdom is endowed with one and the same group, which was transferred from plants to animals and from one animal to another, one group which is the first and foremost, which I therefore still wish to call a protein, a word derived from the Greek proteos, of first rank, suggested to me by Berzelius. And so I will ask a kind of a challenge question. By the year 1850, when Mulder was active, I would say that 6 proteins had been identified and named. Can you guess them, or can you name them? And if you'd like, feel free to drop that into the chat. And if you want to check it on Google and look at, for example, the Wikipedia entry on the history of proteins, I don't think you'll find it there. I think that it's buried a little bit too deeply. So in any case, I'll leave that as a challenge. I count 6 proteins and possibly 7, depending how you count. So I now want to go to the 1960s. And at this point, the first molecular disease had been identified with the famous glutamate to valine mutation in beta globin as the cause of sickle cell disease. The 3-dimensional structures of proteins were just being solved for the first time. Insulin had been sequenced, followed by globins and cytochromes. The genetic code was solved. We had the introduction of the molecular clock hypothesis by Margoliash and others, describing how one given protein family has a characteristic rate of evolution. And then we have geniuses like Susumu Ono, who wrote his book, Evolution by Gene Duplication, and described the evolution of the chromosomes, including the X chromosome. Motu Komura with the neutral theory of evolution. And so with all that, we have examples from the 1960s I would like to mention now, thinking about what we do today. And here we have the first scoring matrix that I've come across, which is from Emil Zuckerkandl and Linus Pauling in 1965. The sequence from multiple sequence alignment of the first sequence proteins is present with the original sequence on the y-axis and a substituent in a multiple sequence alignment in the x-axis. And this is a scoring matrix where some percent of the time, the aspartate D was substituted with asparagine, and these numbers are filled in, and then the red indicates what appeared to be forbidden mutations that were never observed. And so this was at the earliest days of protein sequencing when there were very few data available, and yet these people thought very deeply about it. And we have Margaret Dayhoff generating a series of PAM matrices where PAM is accepted point mutation. She didn't think that APAM sounded very good, so she called them PAM matrices. And in it, she showed that some protein families, such as immunoglobulins or casein, have a large number of accepted point mutations per 100 million years, and others like histones and some proteins like glutamate dehydrogenase had very few substitutions that were accepted by natural selection over the course of 100 million years. And this led to the Dayhoff scoring matrices, in which for any given substitution, it was possible to argue mathematically what a score should be, what the likelihood is of having a cysteine substituted by a leucine, or any pairwise combination. And so why my last thing on history is to give a quote from Zucker, Kendall, and Pauling that will lead us up till today. Life is a relationship between molecules, not a property of any one molecule. So is therefore disease, which endangers life. While there are molecular diseases, there are no diseased molecules. At the level of the molecules, we find only variations in structure and physicochemical properties. Likewise, at that level, we rarely detect any criterion by virtue of which to place a given molecule higher or lower on the evolutionary scale. Human hemoglobin, although different to some extent from that of the horse, appears in no way more highly organized. Molecular disease and evolution are realities belonging to superior levels of biological integration. There they are found to be closely linked with no sharp borderline between them. The mechanism of molecular disease represents one element of the mechanism of evolution. Even subjectively, the two phenomena of disease and evolution may at times lead to identical experiences. The appearance of the concept of good and evil, interpreted by man as his painful expulsion from paradise, was probably a molecular disease that turned out to be evolution. Subjectively, to evolve must most often have amounted to suffering from a disease. And these diseases were, of course, molecular. And I'll remind you, this is the year 1962. And the connection to today is that much of our work in interpreting the significance of variation in DNA sequence and protein sequence, we can trace directly from this deep thinking in the 1960s. We can obtain multiple sequence alignments of any protein family of interest. In this case, some histones. We can immediately identify individual amino acid changes which are divergent, and we can assess the probability of likelihood of heterozygous loss of function changes, or PLI, for example, in the NOMAD project for any given gene. And so most of the models we have to interpret whether variation is deleterious or not comes from these evolutionary paradigms. I'll give you one final slide in this section, which is from the paper by the NOMAD Consortium introducing the NOMAD project. The title of their paper was "The Mutational Constraint Spectrum Quantified from Variation in 141,000 Humans." So this one slide I want to show you has examples of what we can learn. These are Loeff deciles. Loeff is like probability of loss of function intolerant, or PLI, where a Loeff score that's very small corresponds to a PLI that's very high, and a likelihood of being involved in genes that do not tolerate substitution or mutation. And when we look here, we can find on the y-axis the rate ratio for de novo variants in intellectual disability and developmental disorder cases compared to controls, with a 15-fold enrichment of genes that are highly conserved with low Loeff values in the intellectual disability category. So we learned this from the study of variation across 141,000 individuals. The next panel shows partitioning heritability enrichment, where we now focus on minor allele frequency greater than 5% variance. So we're not looking at rare penetrant variants, but at common ones. And we can see almost in a linear fashion, the ones that are very well conserved and have low Loeff scores have a higher heritability enrichment and probably are more important in disease. And so this again is the kind of observation that can only come out from this large-scale sequencing, and again depends on the evolutionary models. And the final one on the bottom, conditional enrichment, over 650 traits were assessed by the UK Biobank and others using GWAS. And these traits include bipolar disorder and schizophrenia, in which again, looking at common variation in per single nucleotide variants and looking at heritability, in this case using regression of linkage disequilibrium scores, we see a pronounced increase in schizophrenia and bipolar, indicating a role for these variants in common disease. And so I mean to say that when NOMAD was introduced, it's useful as we look up one gene at a time, but it's also useful to discern principles by which conserved genes have important roles in disease, whether common or rare. Should I stop for questions or should we go on? I'm happy if someone, I don't see the comments from my slide view. So if somebody wants to ask a question, please feel free. And if somebody has figured out the 6 or 7 proteins, then by all means, let me know.
So we have no takers yet on the 6 or 7 proteins. I'll let you know.
Okay. The report of the Council Work Group on Genomics was championed and developed and led by Thomas. And it's just such an important moment in NIMH genomics to articulate what the recommendations are in terms of funding priorities. And this comes from a panel of experts within NIMH and from the community who suggested what priorities should be. I'd like to show you a handful of slides describing what these main recommendations are. The first is to apply appropriate statistical methods and adopt rigorous significance standards for disease association. This is necessary to interpret both discovery of common or rare genetic variation, and to justify biological follow-up experiments to understand the functional roles of gene variants. And so this is a very important thing. And we need to have this level of strict respect for statistical significance to avoid false positive results. For GWAS, this typically involves criteria for genome-wide significance of p-values smaller than 5 × 10⁻⁸. For rare coding variants, this includes mutation burden tests with family-wise error rates. And so this is something which is very fundamental to thinking about importance of genes. And to go along with this, we should abandon candidate gene approaches in favor of well-powered, unbiased genetic association studies. And so many investigators have a favorite gene, and they feel it must be important. And that feeling is not enough. We need to have candidate genes defined as selected based on unbiased genome-wide approaches and not hunches. And so this is an important direction as well. The idea is that many candidate genes have later proven to be false positive. I'll mention that in some areas of genomics, we tolerate false positives. One example is gene expression profiling by RNA-seq, where we apply an FDR and in some cases accept a 5% or 10% FDR or even 20% FDR sometimes. We say that we can tolerate those because they may lead to biological insights. In the context of gene discovery, we do not want to tolerate false positives which lead to very expensive projects being funded, which do not actually study what they propose to study. I would like to mention some kind of change that's occurring. Sometimes we can have candidate genes selected, for example, in a paper by Evan Eichler's group that came out on Friday on bioRxiv, and a related paper from his team that came out in 2020, in which 16,000 individuals were studied, or 45,000 trios are studied in order to identify mutations in a pre-selected list of candidate genes for which family-wise error rates can still be applied. And these rigid tests of statistical significance can still be applied. We want to further have considerations in selection of variants, copy number variants, exome sequence, common variants, rare mutations. We want to explore all types of genetic variation. This report came out in 2017. Now in 2021, more kinds of technology are available. We can see more types of complex forms of variation. What if underlying schizophrenia or autism spectrum disorder, it turned out that circular RNA was very important, or ring chromosomes, or some kind of genomic change, which is not so traditional, that still might be very important. We want to identify any kind of variation which is conferring risk for disease. Expand genetic association efforts beyond DSM. The DSM provides categorical definitions of what how diseases should be classified. and that there are also ICD codes, for example, in which we say a person does or does not have a particular diagnosis of autism spectrum disorder or something else. And the RDoC project initiated by NIMH includes RDoC domains or research domain criteria, which represents one way to think beyond simple yes/no classification for disease, and try to understand in more depth how we can look at genes, molecules, cells, and circuits all the way to behavior. This particular graphic also shows the environment and neurodevelopment. Capture genetic and phenotypic variation across diverse human populations. NIMH is very active in this area and has included many large studies of diverse geographic ancestry. One example is the Ancestral Populations Network, another the Accelerating Medicines Partnership, or AMP Schizophrenia, in which there are 32 different sites around the world for which early diagnosis of psychosis is attempted, and then following outcomes across different geographic populations. Another main recommendation is to develop and share resources, and we do that heavily through NDA and the NIMH repository and genomics resource. And so this is a large part of the mission of NIMH, to collect and share these data. And samples require robust genome-wide significance in selecting genes. That's similar to the earlier one that I mentioned, but it raises the question, what do you do when there's a GWAS locus that's genome-wide significance, and we might need fine mapping to identify specific genes within a significant locus. And so this is an ongoing issue to know what is the exact variant that we're trying to study and what is the most relevant gene that we're trying to study. Carefully choose experimental systems, and this includes what is sometimes called animal models. We want to avoid saying that we have an animal model for autism when we study, for example, a copy number variant in a mouse model or a gene knockout in a rat model, whatever it may be. So these are some of the recommendations. And again, I'll be happy to pause for any questions or conversation, or we can just wait till the end. Either way is fine. So the final part of the presentation is to think about rare and common disease, and I want to focus on two diagrams. One of them is from genotype to phenotype, and it's shown here at the top. We have DNA to RNA to protein following the central dogma, the cell, the organ, the individual, patients, and populations. Another diagram introduced by Terry Manolio and colleagues in 2009 and in an even earlier paper shows allele frequency versus effect size, looking at very rare to common alleles and essentially the architecture of genetic architecture of disease. And so I'd like to introduce these two graphics. And for the first one, I'd like to mention right away how incomplete it is. It does not include many other steps here, for example, from proteins to protein-protein interaction networks to circuits within a cell and connections of circuits between cells. And so this is still meant to show a general flow from genotype to phenotype. And this diagram also does not specifically, explicitly show changes across development, changes in the environment that affect any of these steps, or changes in disease that we want to compare on this genotype to phenotype continuum. Still, we'll go ahead and say there are different kinds of technologies that can be approached. For example, from genomics to epigenomics, transcriptomics, metabolomics, identifying different kinds of compounds that are present in cells with and without disease, proteomics, biochemistry, organoids. So there are many different techniques here. And then from phenotyping, we're interested in affective and cognitive measures, categorical and dimensional measurement of disease. Neuroimaging, electrophysiology are examples of kinds of phenotyping that we can do. We want to apply this kind of thinking across the disorders that are in the realm of NIMH, from autism spectrum disorder to bipolar disorder and depression, schizophrenia and suicide, and many others. At the same time, we're also interested in patient engagement, patient registries, ethical and legal issues, clinical trials that are ongoing, and how to connect our genomics all the way to therapeutics. So this is kind of an outline of a genotype to phenotype continuum. And on it, we might take the example of comparing the phenotype of autism spectrum disorder and comparing it to neurotypical controls and ask what genes are there. For example, are there genes for autism spectrum disorder? And many neurodevelopmental and psychiatric disorders are highly heritable. We want to understand the nature and extent of deleterious genomic changes, for example, in ASD compared to control. And what does it mean to say we want to find a gene for something? For that, I'd like to refer back to a paper from Ken Kendler published in 2005, describing possible genotype-phenotype relationships. There may be a one-to-one relationship or one-to-many. There may be a many-to-one relationship. For example, one might think of mutations in different single genes that can each cause a single disorder. I think of, for example, Cornelia de Lange syndrome. Or for one-to-many relationships, we can think of mutations in MECP2 leading to entirely different phenotypes in boys and girls. And then there may be many-to-many relationships. And so Ken Kendler went on to talk about what it means to identify a gene for some trait or psychiatric disorder, noting 5 criteria. And by the way, the reference is given below for those who would like to see the paper. But the strength of association of X with Y may be quantified, for example, by an odds ratio. We're interested in the specificity of a relationship of X with Y, as shown in the previous slide. Is there a direct connection between the mutation in a gene and the phenotype? Non-contingency of the effect of X on Y. Is there a third gene or factor, such as the environment? In this case, I just drew a Z and crossed it out. Say, is there a factor Z, which also influences the effect of X on Y, which would make it more challenging to describe it as a gene for some condition. The causal proximity of X to Y, and the degree to which X is the appropriate level of explanation for Y. And Ken Kendler's conclusion was, we do not have a gene for autism or a gene for a particular neuropsychiatric condition. I'd like to take a couple minutes and talk about 3 examples from my own work in the past in which there is a direct connection. The first one is Sturge-Weber syndrome. And in this, there's a port wine birthmark that occurs in about 1 to 300 people, as shown here in this newborn who unfortunately died from this condition. A subset of people with port wine birthmarks have Sturge-Weber syndrome, which is characterized by features seen on MRI, such as enhanced leptomeninges and atrophy of the cerebral cortex. And so the atrophy is seen kind of pinched in here on this MRI on a whole hemisphere ipsilateral to a birthmark. And so it had been hypothesized by Rudolf Hapel and others that conditions that cause neurocutaneous disorders such as this might be caused by mosaic variation. And so we went ahead and obtained genomic DNA from a birthmark directly and from a presumably unaffected part of the body, sequenced those genomes and compared them, and identified a single base change that was somatic and mosaic in GNAQ as the cause for Sturge-Weber syndrome. And in this case, there is no GWAS. There's not a large cohort. There were 3 probands. And after finding this in 3 probands, we followed up with some dozens of samples, and other groups in Japan and in Europe and in the United States repeated the study and found the identical result, that this mutation in one base pair in GNAQ is directly a cause for Sturge-Weber syndrome. So in this case, the gene GNAQ encodes the G protein alpha subunit, which is coupled to a 7-transmembrane receptor. That leads to persistent slight activation of downstream signaling pathways, such as phospholipase C beta and ERK, which is phosphorylated abnormally in this condition. An interesting thing is that this picture here from Silvio Gutkind and colleagues from a review article about intracellular signaling pathways includes many genes involved in signaling, which when mutated cause neurodevelopmental disorders, TSC1 and 2 in tuberous sclerosis, NF1 in neurofibromatosis, AKT1 in the Proteus syndrome, PI3K in Clove syndrome, and we added GNAQ to the map with Sturge-Weber syndrome. And so the other thing that I find interesting is that these same genes tend to be tumor suppressors or oncogenes in the world of oncology. And so they cause cancer. And at a different time and place, they cause these neurodevelopmental disorders. And so this realm of work is quite distinct from seeking genetic contributions to schizophrenia, because these tend to be single gene disorders and rare disorders. And so I think it's very important to think about the difference between those kinds of conditions and the kinds of mutations that occur. A second example, in work from David Goldstein's lab, I contributed to this from the title, Whole Genome Sequencing of a Single Proband, together with linkage analysis, identifies a Mendelian disease gene. This happened to be PTPN11 in metachondriomatosis. And so again, you wouldn't tend to argue that one has found a gene for schizophrenia by doing linkage in a single pedigree, although that has happened, come to think of it. And yet here, with a Mendelian disorder that's rare and somewhat penetrant, but not completely penetrant, we could argue that we have identified a mutation in a gene from simply, in this case, 2 pedigrees. The third case comes from a collaboration with Kun Shah and Evan Eichler, and there are about 75 co-authors, and I'm in there with a graduate student somewhere in the middle. We found, as a team, 17 children around the world who had de novo loss of function mutations in the gene CSDE1. And each of these probands had a flavor of autism spectrum disorder with similar dysmorphism, similar phenotype, not identical, but similar. And that there was a Drosophila and a mouse model that we reported as a group. Again, I don't want to take credit for leading this project. I was just a small part of it. But there were defects in synapse, axon outgrowth and synaptic transmission in both mouse and Drosophila models. And so you have an example here of a very rare form of autism with very few probands in a gene which has mutations with a very rare allele frequency. And yet it can teach us something about autism in general and idiopathic autism, potentially, if we collected, for example, 100 examples like this and found that many of them involved, as in this case, a gene encoding an RNA binding protein, in other case, a gene encoding a protein implicated in neurotransmitter release. We might build up models of biological pathways which are perturbed in schizophrenia in general, or in autism spectrum disorder in its idiopathic general sense. So coming back to our picture, we can think about one gene or several genes connected to a specific phenotype in rare disease. And then we can think about common disease with GWAS, when we might want to gather 10,000 or 100,000 individuals with and without the condition. What we expect to find with GWAS is common shared genetic variation that confers increased risk. We expect common variants to be tolerated evolutionarily, to not be penetrant in their effects, so they have small effect sizes. And this takes us to Terry Manolio's figure, in which she and her colleagues plotted allele frequency versus effect size and describe visually genetic architecture of disease, with rare alleles causing Mendelian disease. They're rare on the x-axis with high effects on the y-axis, and then common variants implicated in common disease by genome-wide association, with an expectation that we would not have many examples of high effect common variants, and that we would have an especial challenge to find rarer variants of small effect size, although they should exist. So genetic architecture in general is characterized by how many genetic variants influence a phenotype, the population frequency that's on our x-axis, the effect size on our y-axis, and interactions of these genes with each other in the environment, which are also challenging to measure. Rare variants we can expect to be often de novo with large phenotypic effects, often Mendelian. They may have variable expressivity. And a gallery of examples. In breast cancer, we see the basic shape of that plot from Terry Manolio and colleagues, with the rare variants indicated here to common. Colorectal cancer, we find the same kind of pattern with lower effect size genes identified that are more common. Diabetes, with type 1, we do have these rare penetrant variants. With type 2, we have not found them. And so these authors, Timpson et al. In this cited paper, argue that distinct genetic architectures influence our opportunities to create diagnostic tests and for biological validation of therapeutic targets. And so we're very interested in understanding genetic architecture. For Alzheimer's disease, a similar profile. And again, I intend this just to be a gallery to say that we see this same kind of shape across different diseases. And now to schizophrenia, where a paper from Duncan et al. Is showing that there's an upper bound on the effect sizes helped along by natural selection, and that what we can see here in orange is results from GWAS with 80,000 individuals, and that when we double that and go to 160,000 individuals, we tend to see lower effect size variants, which are still common. And so what can we expect as we move up to more and more hundreds of thousands of individuals in GWAS? The final slide on this kind of plot, which I'll show you in several forms, is the schema paper that has come out in medRxiv as a preprint. Again, the reference is down below, showing minor allele frequency in the general population versus odds ratio. The common effect genes are shown here on the x-axis, and then the rare variants here, consisting of both the protein truncated variants in red and the copy number variants in green. So in preparing to conclude my comments, I want to just raise a couple questions. What's going on in the middle? Why is that empty? And perhaps you have answers for that. What is the relationship between the functions of these genes that are rare and penetrant and the functions of these genes that are common and not so penetrant? Do they actually have a functional connection? What about the copy number variants? When we look at 3q29 deletion, for example, individuals with that have an extraordinarily highly elevated risk for psychosis. What are the individual genes within the 3q29 deletion region that are most important, or elsewhere in the genome perhaps? And so there are many questions raised by this kind of image of the architecture of these diseases. I'd like to conclude then. At NIMH, we support the discovery and interpretation of variation associated with neuropsychiatric conditions. And we're interested in the genetic architecture of disease as part of this mission. A great challenge is to predict the functional consequences of genomic variation. Progress in understanding these genomic contributions may lead to improved diagnosis, prevention, and treatment. We're very interested in the connection between genomic variants that confer risk for disease and therapeutic efforts. And that is a very large challenge for sure. We support the development of data resources, technologies, and bioinformatics capabilities and their application to disease. The final slide is NIMH envisions a world in which mental illnesses are prevented and cured. And our mission is to transform the understanding and treatment of mental illness through basic and clinical research, paving the way for prevention, recovery, and cure. Now I'm clicking escape on my PowerPoint. I'd be delighted to have a conversation or questions or comments.
Thank you, Jonathan. Thanks for laying out in a fascinating way the challenges, the progress and the challenges that we have made in the field. Right? So first thing, go ahead.
No, please, after you.
The first thing comes to mind is I could think of a number of projects that could, try to answer some of these questions, but they would all be very expensive given current technologies. What's the appetite? And it's sort of tacky to start with money, but what's the appetite by NIMH for these, high impact, high budget projects?
So I've been an academic my entire career, and I was very happy being an academic until actually NINDS and NIMH called me both and said, would you please come in? And so I came to NIMH, and I've only been there a few months. But one thing I've learned is to be very careful with my words and not to tell you anything that would be unfair to others. And so if I told you about initiatives and the amount of money we would put to do a certain task, I would want to tell everyone equally and not give any advantage to this group. Not because I don't want to talk, but because I'm having it drummed into me to be very fair.
Yeah.
And so that's why I'm going to be very cautious in answering. And so my caution is, actually, I could say this. I believe there are things I'm not allowed to say to you, but you are allowed to say to me. So you could tell me what you think. If you had unlimited money, what would you do? And I will take notes.
So, the purpose was not to put you in a difficult position, of course, right?
Not at all.
But practically, if you start from the beginning of your remarks, these sequences, so the sequencing studies, for example, right? So they are very difficult to conduct at the scale and the power that is necessary, right? So the reason why folks are still doing exome studies or have now defaulted to low power sequencing studies is really because we can't, besides some technical challenges with the analysis of the data, is we feel the field feels that it's not affordable with current technologies, though it is almost necessary to do at some point because we won't find all the answers otherwise. And you mentioning long read sequencing is a great example also, right? Because there are some designs that we have considered that would require long read sequencing. And again, as long read sequencing is still fantastically expensive, it is also very difficult to scale up. And I don't expect you to have answers to these challenges, but this is something that, the investigators that I work with in the field face all the time, right? We want to
Here's
put
what
together. Go ahead.
I think that the first person to sequence 10 genomes from individuals diagnosed with schizophrenia relative to 10 controls by long read sequencing at a cost of $10,000 per genome will have spent a lot of money. And what if 6 or 7 of those 10 genomes from individuals with schizophrenia have recurrent, penetrant, structural variants that are invisible to other technologies?
Right.
I think the whole field would say, why didn't I do that sooner? Because it was really important. And do I need to have a sample size of 10, 100, 1,000, or 10,000 to make discoveries? It depends what you're looking for. And so I would not be surprised if there were a phenomenon that is simple to discover that we've all missed. And that in 5 years, we look back, and one example that comes from colleagues at Johns Hopkins is finding protein aggregation in postmortem brain samples from individuals with schizophrenia. So there, you can look in the brain banks, you can see the brains, and you can find that this aggregation happens in cases and not controls. That would be an example of something, if it were true, that is fundamental. And so I mentioned earlier ring chromosomes or circular RNA or gene fusions. There might be simply structural variants that are easy to understand, but have not been detected. And so I feel that the sample size of 1,000 or 10,000 will be very impractical in the short term because of cost, but that a small cohort could be highly informative still. And some people are saying, I want to go forward in a series of different projects. Those people will make the first discoveries, and I will listen to any argument from any investigator who says, you should let me try something high risk. I'd like to hear it.
I hear you, right? What I seem to hear is that you're very interested to answer some of the questions that can be answered by very targeted study designs, even if they use technology and by using technologies that are not currently scalable because of economic considerations. And that is a great message to get. There's an additional complexity that, as someone who is now on the other side of this divide can ask you, while this may work for you scientifically, it's of interest programmatically, there's another challenge for the field then to pass it through peer review. And here, of course, the solution is to targeted investments, aka RFAs or similar mechanisms, right? Where you can send a message to reviewers on how you want the projects to be looked at scientifically. So without these going hand in hand, I think it would be very difficult for the field to do this at a scale that would sort of move the needle on progress.
So I agree with your comments. I am working actively on RFAs. I just can't say which in the spirit of fairness, but that it's important to do what you're saying and have a mechanism to tell the field, this is really a big pot of money to do something that otherwise might be too difficult to try to achieve. And I think there are many choices in how to do that. And when you just made your comments, you used the word targeted. That could mean targeted in terms of technology, or in terms of design and a limited sample size. We never want to encourage people to do an underpowered study. And we only want the most rigorous science. But you're bringing up the point that the field has massive unanswered questions. They're enormous questions we don't understand, and that there are choices in how to go. One example is to take GWAS up to, I'll pick an arbitrary number, 800,000 exomes. A different answer would be take it to the first 5,000 whole genomes. A different answer would take it to the first 500 telomere to telomere genomes. And so there are choices that need to be made, and I don't have the answers, but that's part of my new job, is to try to listen to the field. Listen, I'm here to talk to you, but also to listen. We only have till 5 o'clock. I can stay longer, but I assume that people might have to go off. But that I hope this is the start of a conversation, and I welcome Zoom meetings with any of you one-on-one or with a group again to continue the conversation if we need more discussion time. I'm very eager to communicate with you and hear more about what your ideas are.
I thank you, Jonathan. Since I seem to have dominated the conversation, I want to hear if anyone else.
Yeah, I think Dalton and I have our hands up. Dalton, you want to go first?
You can be my guest if you like to go first. Go ahead.
Well, okay, mine's brief, but you know, and what your comments in response to Thomas corresponds to what you showed on the slide of the schema results, which is, it's my sense as a non-card carrying geneticist, but sort of an end user, that the trend in the field had been increasing the sample size to have ever greater power to be able to look for the small effects of the myriad number of genes that may be susceptibility genes, or to expect ongoing improvements in the technology and greater resolution and sequencing to reveal genetic factors that previously were invisible to be detected and indicate genetic mechanisms of heritability. But the middle zone, as you show, which is kind of less populated, reflects that it may be that we need to examine targeted phenotypes, enriching the sample that's assessed in terms of the specific type of phenotypes. And that's an approach that hasn't been used commonly previously, but I just wanted to know what your thoughts about that. And that reflects or relates very much to what Thomas was saying, is, you know, running the gauntlet of peer review without having some kind of protection provided by, you know, solicited applications or contracts.
Great question, and I thank you. I think that there may be variants in that zone between the common and the rare that are now called variants of unknown significance, and with further phenotyping, they will be elucidated. NIMH is supporting rare gene discovery efforts, and it's also, of course, promoting GWAS with ever larger sample size. So I don't know of an RFA that specifically targets the middle region. And I would say that there's tremendous interest in expanding how we think about phenotyping and going from categorical to dimensional. And that may lead to more discoveries, even in the reanalysis of existing data.
Thanks, Jonathan.
So one more question.
Sure.
Unfortunately, we'll have to end after this, so I have to go. There was
Do you
another.
have time for my question then?
If you make it brief.
Yeah. I'm interested in whether or not you're seeing a lot of studies being proposed on social genetic effects or genetic nurture effects, given these phenotypes are highly sensitive to, for example, household environment, and in a lot of the genetic correlations have been shown to be actually happening at the indirect genetic effect level and not the direct, with the exception of autism in a recent paper in PNAS. So, but for example, depression, schizophrenia, all of these, part of what we're detecting, at least with the common variants, is actually the indirect genetic effects through the immediate family environment. Is that something that NIMH is interested in, or are you seeing that as outside the purview of your program?
So the 30-second answer is that NIMH has a tremendous interest and commitment to understanding social effects. And there are different divisions within NIMH, such as DTR, the Division of Translational Research, that has a more focused interest in that. The genomics portfolio per se has been more on the basic science side of the spectrum, but this concept is really important and we would be open. I'll take the last 10 seconds if I have that, Thomas.
Sure.
To say three things. Number one, yes, I'd be glad to share the PowerPoint. Number two, I want to repeat that I'm glad to continue the conversation with anyone. Please just call me and we'll Zoom or however you'd like to meet. And number three, albumin, casein, crystalline, fibrin, gluten, hemoglobin, and pepsin.
Thank you. Thanks for that. And we will invite you again if you'll join us maybe in a year or so. I want to thank you. I want to thank everybody and wonderful.
Hey, thanks for coming.
Bye-bye.